Thank you very much, Sol, for agreeing to give a Build a Cell seminar, and you can get started.
Okay, thank you, Kate. Thank you for the invitation to give a talk. Okay, so I'll start by just introducing myself. I'm Saul Purton. I'm a professor of algal biotechnology at University College London, which is based in the centre of London. The picture you can see there is the original building of the college that was opened in 1826. So we're just coming up to our 200th year anniversary. In terms of my background really is as a molecular biologist. I did my first degree in biochemistry at the University of Bristol, PhD in Cambridge in plant molecular biology, and then postdoced in Geneva in Switzerland for a few years, working on algae before joining the faculty of UCL, and I've been there ever since. And I've been working on algae ever since. So what I'm going to tell you about today is some of the work that we've been doing, exploring how we can exploit single-cell algae as platforms for making recombinant products, so products of potentially commercial value, sort of exploring how we can use microalgae as a light-driven cell factory. And in particular, focusing in on the algal chloroplast. So just to give you a little bit of context, I can just try and find my, there we go. So as I say, microalgae offer the potential for making high-value products using light as an energy source. So a very simple, low-cost cell factory where your inputs, and I'm just going to switch to the laser pointer. Here we go. Your inputs are carbon dioxide and basic inorganic nutrients, and the whole system's been driven using light. And one of your outputs, obviously, is oxygen. So it's very sustainable technology, and there are a number of different natural products that are being produced commercially using algae. So edible algae can be used, obviously, as food and feed ingredients. So you can use the whole algae as a replacement for animal proteins, as well as a sort of highly nutritional component in food and feed. There's a lot of vitamins and minerals and things in the algae that are beneficial. And then other natural products such as biopolymers for making sort of bioplastics, oils for making things like biofuels, various different metabolites that have bioactive properties. And there's a huge amount of work being done in terms of exploring the potential of microalgae to produce novel bioactives. And then obviously algae are pigmented, so their pigments are used for light capture, but those pigments can also be used as colorants and dyes and inks. So a wide range of natural products that can potentially come from algae. And I should say that there are hundreds of thousands of different species of algae, both marine and freshwater. So this is very much an untapped or poorly tapped space in terms of exploiting and exploring different algal species. And then, as I say, our interest is whether we can use genetic engineering approaches to engineer these algae to make new things, so different recombinant products. And that could be anything from recombinant DNA or RNA that may have therapeutic applications, different proteins, enzymes, and other sort of biologics, or making novel metabolites that are of value. And actually, for both of these different applications, then there is a need to develop sophisticated genetic engineering tools. And even if you're producing a natural product, most of the algae that's grown commercially is essentially, or are essentially, wild isolates. So very much like the start of agriculture, where the crops that were being grown were very sort of weedy, early sort of ancestors of the highly domesticated strains that we have today. Then there's a need to take these wild isolates of algae and to make them more robust for intensive cultivation, as you see here. And then obviously we need to develop these tools for making these recombinant products. So what I'm going to tell you about today is a little bit of our work where we've been developing these tools for the chloroplast, or engineering the chloroplast of one particular algal species. And that algal species is Chlamydomonas. Chlamydomonas is a single cell freshwater green alga. You can see that it's motile, it has 2 flagella. It has a single chloroplast that fills about half of the cell volume. It has an eye spot within the chloroplast that allows it to detect light direction, and therefore it can swim towards or away from the light. Why Chlamydomonas? First of all, it's probably the most extensively studied single-cell alga. It's been used for over 60 years or so as a model for studying things such as photosynthesis and cell motility, principally because it's very easy to cultivate. It grows relatively quickly, so doubling times of sort of 6 to 8 hours. It's perfectly harmless. So it's in the US, it's already recognized as a grass organism, generally recognized as safe. And because it has a haploid nuclear genome, then it's very easy to generate mutants because those mutations are not masked by additional copies of the mutated gene. So there are large mutant libraries. And one of the nice things about Chlamydomonas is that it can dispense with photosynthesis. If you feed it acetate as a fixed carbon source, then you can grow it in the dark and it doesn't need to photosynthesize. And that's allowed the isolation of many interesting photosynthetic mutants. It also doesn't need to swim, so there are lots of flagella mutants, and that's actually allowed Chlamydomonas to be used as a model system to study ciliate disorders in humans, because the flagella of chlammy is essentially identical to the cilia of higher animals. In terms of genetic engineering, then we have very detailed genome sequences for both the nuclear genome and the chloroplast and the mitochondrial genomes. So they've been sequenced and fully annotated. And we have very well-developed molecular genetic tools for engineering, in fact, all 3 genomes. So it's actually the only organism for which there are transformation procedures for 3 different genomes: the nucleus, the chloroplast, and the mitochondria. And for the nucleus and the chloroplast, there are now Golden Gate or Moclode-based assembly methods and parts libraries. And essentially that just allows you to build your designed gene very quickly using a standardized assembly method. So as I say, our focus is on the chloroplast. Why the chloroplast? Well, because it's simple. A lot of the research in my lab is really based on let's keep it simple so that we can do it quickly, easily, and cheaply. And we've focused in on this very small circular chromosome that is the genome of the chloroplast of Chlamydomonas. So it's about 205 kilobases in size. It contains 70 protein coding genes, and of those, 43 are non-essential. I mentioned that we can dispense with photosynthesis in Chlamydomonas, and the majority of genes actually on the chloroplast genome of plants and algae are involved in photosynthesis. So we know actually that we can dispense with over half of the protein coding genes. It uses the standard genetic code, although it doesn't use UGA as a stop codon, and I'll come back to that later in the talk. In terms of translation, it contains 26 tRNA genes, and that is sufficient to translate all of those protein coding genes. It also has various other features that basically just make it even simpler than the equivalent chloroplast genomes from, say, higher plants. So there's no RNA editing, there's no overlapping of genes, there's very few introns, and they're all non-essential. And it just uses a very simple transcription system one type of RNA polymerase with a single sigma factor. And we have a very detailed sort of transcript map of the genome. Importantly, we can do chloroplast transformation. So we can put transgenes into the chloroplast. And that's something that's really only feasible with either Chlamydomonas or a few higher plant or land plant species, predominantly tobacco, but a few other Solanaceae species can be transformed as well. So in terms of doing reverse genetic studies and genetic engineering of the chloroplast, then Chlamydomonas is one of only a few model systems that really have sufficiently developed tools, molecular genetic tools to do that. And importantly, we can target our transgene to any part of the genome. So basically, any DNA we introduce can be targeted into the genome through homologous recombination events that allow us to precisely insert the gene into a predicted site. And if we build the gene correctly, then we can get high-level expression. And the chloroplast, unlike the nucleus, doesn't have mechanisms for gene silencing. So once we've got a gene in and it's working, then that expression is stable. We haven't got to maintain any kind of selection to keep the gene active. And I just wanted to sort of highlight or illustrate, how simple a genetic system this is. Very often when we're thinking about synthetic biology, we tend to think of things like E. coli as a simple platform, a simple chassis for genetic engineering. And I just put up here the genome of E. coli for comparison. So the genome of E. coli, it's an order of magnitude bigger than the chloroplast genome. There are over 4,000 genes here compared to the 100 genes that are present on this Chlamydomonas genome. So compared to even what we think of as a simple synthetic biology chassis, then the chloroplast is an order of magnitude simpler. Okay, so our research really is, we design genes in silico, we introduce them into the chloroplast of Chlamydomonas, and we use the fact that we can genetically engineer the chloroplast either to make various different desirable products, which could be therapeutic proteins or biopolymers or novel RNAs or metabolites, or as you'll see at the end of the talk, we're also using the fact that we can do genetic engineering of the chloroplast to start to sort of explore how we can manipulate this very simple genome in terms of its genetic code. So can we do things like reduce the codon usage down from 63, remember it doesn't use that stop codon, to an even lower number like 51? Can we think about reorganizing the genetic code in the chloroplast? So the chloroplast is this self-contained genetic system. So potentially we can move away from the universal genetic code. And can we start reassigning codons for amino acids beyond the 20 natural amino acids that are used in life? So I'll talk a little bit about this at the end, but I'm now going to sort of focus really on these various applications and making various different products. So in order to do that, we've developed a number of tools, and we call this sort of suite of tools STEP, or SynBio tools for engineering plastomes, chloroplast genomes. One of the things we've developed is some software so that we can codon optimize the coding sequence of any new gene that we put into the chloroplast genome. And the reason for doing that is that the chloroplast genome is very AT-rich. So it actually has quite a strong AT-rich codon bias. So we can optimize our gene or the coding sequence for our gene. As I say, we and others have developed very simple one-step or one-pot assembly methods for putting all the different parts together, the DNA parts that are needed to actually make a functional gene. So that's sort of illustrated here. The blue is the coding sequence. The arrow here is a promoter. The 5' untranslated region with the ribosome binding site, a terminator region. And then these green regions here represent chloroplast elements that allow 2 recombination events that target our DNA into a chosen region of the genome. So we need to put all those parts together to actually make our transformation plasmid. And the technology that we've developed for assembling them is based on using this enzyme SAP1, which provides these bespoke 3 base overhangs. And the reason that we've chosen SAP1 is that basically we can do scarless joining of these different parts. And that's particularly important in the chloroplast, where the context around the start codon, the ATG, and the stop codon can be critical for efficient expression. So we can join any ribosome binding site to any coding region without introducing any DNA scar at these sites. And we can do all of this in one pot. So we can add all of the parts together and ligate them and assemble our plasmid in a single step. So that greatly accelerates and standardizes how we build our constructs. Another thing that we've developed, and again, I'll talk about this a little bit later, is a strategy for biocontainment, whereby we can put that unwanted or the unused stop codon, TGA, in the middle of our coding sequence. And then we can suppress its function as a stop codon by expressing a tRNA gene, in this case for tryptophan, that will recognize that stop codon as a tryptophan codon, and therefore we get full translational read-through. So this allows us to have a level of biocontainment of our transgene. Essentially, if this gene escapes into another microorganism, then our prediction is that it wouldn't be functional because of those stop codons. Another consideration in terms of the sort of regulatory considerations around genetic engineering of microorganisms is to avoid using antibiotic resistance-based markers for transformation. So we have developed methods whereby we exploit the fact that, as I say, Chlamydomonas can dispense with photosynthesis, provided you give it a carbon source such as acetate. So we can use that fact as a way of selecting for transformants. So in this example here, we've got our gene of interest, and then one of the flanking regions contains a photosynthesis gene that's critical for photosynthesis. If we introduce that into a mutant where we've disrupted that photosynthesis gene, then we can restore that photosynthesis gene with the DNA carried on the plasmid, and at the same time introduce our gene of interest. So we end up with a photosynthetic transformant where the only thing we've introduced is, or the only foreign DNA we've introduced is the gene of interest. We've also developed a system for serial transformation and scarless loop out of regions of the plastome. And this basically just exploits the fact that, as I say, homologous recombination occurs readily in the chloroplast. So if we wanted to delete this yellow gene here, for example, we can build a gene construct that looks like this, where we've got this double marker here. So AADA is a marker that confers spectinomycin resistance. COD-A is a negative marker that confers sensitivity to a compound called 5-fluorocytosine. If we build a construct where we define the DNA we want to delete, with the right-hand side being this flank, the left-hand side being this short repeated region that we build into the construct, then we can select for transformants that have replaced the yellow gene with the double marker to create an intermediate transformant. And then, because this is now sensitive to 5-fluorocytosine, we can use that fact to drive recombination between these duplicated direct repeat copies that flank the marker, with the net effect that we end up looping out this whole cassette and we end up with a completely scarless, perfect deletion of our target gene. So that tool allows us to do very clean excisions of sections of the chloroplast genome. Okay, so we've got a sort of suite of tools, so we can very quickly design and build any gene of interest. We can introduce it into our target strain using either a gene gun, so-called biolistic transformation, where we take the DNA, we coat it onto gold microparticles, and we bombard a lawn of Chlamydomonas cells to select for transformants. Or there's a poor man's option, which is essentially to take a suspension of the cells, the DNA, and some glass beads, and just using a lab vortex, just mix the whole thing together for about 15 seconds. And the abrasive action of these glass beads essentially creates transient holes in the cell and in the chloroplast membrane, allowing the DNA in. It's not very efficient. This is a glass bead transformation here. You can see that probably about 1 billion cells are being treated, and we've probably got a handful of transformants. But in fact, that doesn't matter, because in principle, each of these independent colonies should be genetically identical, because the DNA is always predicted to integrate at a precise site in the chloroplast genome. So we can pick off a handful of those colonies. We can do a very simple PCR screen just to make sure that the DNA has indeed gone in at the right site. And this particular PCR setup is designed to test both correct insertion and what's called homoplasmy. Because one of the things I didn't mention is that the chloroplast genome of Chlamydomonas is present in multiple copies, anywhere between 40 and 80 copies per cell, depending on the growth conditions. And so we want to ensure that every copy of the chloroplast genome has acquired the transgene. So here in this PCR screen, we use the absence of this lower band here. So these are controls before transformation, to show that we've lost that original copy or those original copies of the chloroplast genome, and that all of the copies have the transgene. Once we've confirmed that, which takes a week or two, then we can check that the gene's actually being expressed. So we can do protein analysis if we're producing a protein. Typically, we put an epitope tag onto the protein, so we can use a commercial antibody to show expression. The expression levels we get are not as high as you might get in, say, E. coli or yeast. Typically, they're below 1% of the total soluble protein. So it's hard to see bands on a stained gel, but using an antibody, you can clearly see that we're producing the protein. Once we're happy with that, then we can scale production. So these are, I think they're 15-liter bags. This is our simple little photobioreactor setup in our lab with just using artificial lighting. We can scale up, and then we can harvest and dry the algae. So just give you some examples of things that we've made in Chlamydomonas. One of the first things we tried was human growth hormone, essentially because it's a nice, simple, small protein, no cofactors, and it's recognized as being a significant commercial protein. Not sure what the current market value for human growth hormone is, but it's certainly many millions. So we produced that successfully in Chlamydomonas, and we could show that if we just took a crude cell extract and added it to a particular cell line, mammalian cell line, that was dependent on growth hormone for cell division, you can see that when we add the extract, then there's a marked growth of the cell line compared to the controls. So it was a nice demonstration of making a biopharmaceutical in the algal chloroplast and demonstrating that it's functional. Some other enzymes that we've focused on and where we think there is real opportunity for sort of commercialization are phage endolysins as antibacterial enzymes. So these are viral proteins that are produced by bacteriophage that infect a specific bacterial pathogen, and the final step in the infection cycle is for the infected bacterium to be lysed, and that's brought about by the endolysin, which breaks down the cell wall of the bacterium, the peptidoglycan cell wall of the bacterium, and releases the progeny phage so that they can start the cycle again. And these enzymes, as I say, they're encoded on the phage genome. They're very simple enzymes, single subunits, no cofactors. The enzymes are highly specific for the particular bacterial species because they recognize a key feature of the peptidoglycan cell wall. These viral enzymes have co-evolved with their bacterial targets, so there's actually very low emergence of bacterial resistance to these endolysins. And if you produce these endolysins recombinantly and add them to bacterial culture, then they will break down the cell wall and cause lysis of the cells. So cells are under a turgor pressure, they will burst if you compromise the cell wall. So there's a lot of interest in using these phage endolysins as antibacterials, as an alternative to conventional antibiotics. The key problem is the cost of production. So if they can be produced in a very cheap platform such as algae, then potentially we can produce these endolysins and commercialize them. So as I say, these endolysins will target various different aspects of the peptidoglycan cell wall. In gram-positives lysins, these are more effective simply because gram-positive bacteria don't have a protective outer membrane protecting the peptidoglycan cell wall. So most of our focus has been on gram-positive lysins, but there is promising work being done on gram-negative lysins as well, getting them across the outer cell membrane so that they can target the peptidoglycan cell wall. So one example, we've produced 2 endolysins against Streptococcus pneumoniae, and again confirmed that they're produced in Chlamydomonas, in the chloroplast, and again shown that we can just take a crude algal extract, add that to a culture of the Strep pneumoniae, and by monitoring the optical density over time, you see this drop in OD. So this is basically the bacterial cells lysing or bursting, and essentially after about 80 minutes, all of the bacteria have completely lysed. So this is, as I say, it's just a crude extract being added to a dense culture of the bacterium. So very effective as an antibacterial treatment. The green line here is basically a chemical treatment that also lyses very effectively the bacterium. So you can see that if we take the treated bacteria after 80 minutes that have been treated either with PAL or with the other endolysin CPL1 and plate them out on growth media, then we see none or almost no colonies arising. So complete killing of the bacterium over an 80-minute period. So we're exploring lots of different applications of this, everything from sort of treating skin diseases, bacterial infections of skin diseases, to aquaculture and agriculture, where bacterial pathogens are a major problem, and a problem that's usually treated using sort of indiscriminate antibiotics. So we're exploring whether we can actually use algae as an alternative. Another example of an application is producing subunit vaccines in algae for aquaculture. specifically in this case for shrimp farming. So shrimp farming is a major industry in Southeast Asia. So we're collaborating with groups in Thailand to look at mechanisms for protecting shrimp farms against infection with things like white spot syndrome virus. So if this virus gets into the farm, then it completely devastates the crop. And obviously you can't vaccinate tiny shrimp through injection to protect them against viruses. They're too small and the cost is too high. But what you could consider is actually having an oral vaccination system whereby you formulate the vaccine into the feed. So because Chlamydomonas is edible, but perfectly safe, the idea is that we produce the vaccine in Chlamydomonas, we dry the algae, we formulate that in the feed, and then we feed that to the shrimp. So this was one early example where we produced the major envelope protein for white spot. The expression level was actually quite poor. The upper band is a control band, and you can see the VP28 band is actually quite faint. But when we feed the shrimps with the algae, then it has quite a pronounced effect on their survival. So this is a graph that's basically showing the survival, or the which way around are we? The mortality following challenge with the virus. So at day 8, in the absence of the vaccine, we've got almost 100% mortality, whereas those shrimp that received the feed containing the algae and the vaccine, the mortality rate is around about something like 15%. So you can see that this has real potential as a very low-cost, low-tech way of protecting shrimp against a major virus. And we've sort of gone on from subunit vaccines to also look at using double-stranded RNA as a sort of vaccine treatment in shrimp as well. This is obviously to exploit the fact that double-stranded RNA, if taken up by the shrimp, can actually elicit a gene silencing response through a mechanism called RNA interference. Essentially, the double-stranded RNA will specifically lead to the silencing of a key viral gene and therefore preventing the infection. And again, the gene that we're targeting is the one for VP28. So we've developed a very simple method for producing double-stranded RNA in the chloroplast of Chlamydomonas. And the chloroplast is particularly attractive as a platform for making double-stranded RNA because there are no enzymes that specifically degrade double-stranded RNA, unlike in bacteria. So these long double-stranded RNA molecules will accumulate in the chloroplast. So we've developed a very simple one-step method where we can take any target DNA. In this case, it's a section of VP28, but it could be anything. And we drop this into this cassette or into this plasmid and replace this red cassette, this red fluorescent protein cassette. So we have a very simple way of checking which E. coli have got the recombinant plasmid. They're the white ones. And when we introduce this construct into Chlamydomonas, we have transcription from 2 strong promoters, one on the top strand, one on the bottom strand, that produce this double-stranded molecule. And we can demonstrate the accumulation of that by reverse transcription PCR. So again, when we feed shrimp with dried algae that's producing this double-stranded RNA, in this case, we're looking at survival here. And again, you can see that there's a marked increase in survival from about 10% survival at day 8 post-infection to something like 70%. So again, a very cheap, a very effective way of protecting the shrimp against a viral pathogen. Okay, so that's a few examples. I wanted to come back to this observation that we made a few years ago that the chloroplast genome of Chlamydomonas doesn't use one of the stop codons. There's nothing special about that. It's just that, as I say, the codon bias in the chloroplast is very strongly towards AT-rich codons, and so effectively almost all of those 70 protein coding genes choose to use TAA as a stop codon. A couple use TAG, but none of them use TGA. So we realized that actually we had a free codon that we could use for other things because it wouldn't affect any of the endogenous genes. So for example, we could take one of the chloroplast tRNAs and change its anticodon so that it would recognize that stop codon as if it were a sense codon, as if it encoded a particular amino acid. The reason we wanted to do this was actually because we were running into trouble or running into problems cloning some of the genes, particularly the antibacterial genes, cloning them in E. coli. So if we just built a construct like this with a chloroplast promoter, ribosome binding site, and terminator, these elements are weakly expressed or weakly functional in E. coli. So you're going to get some low-level expression of this toxic gene. So if it's toxic to E. coli, then basically it becomes unclonable. So we get full-length translation in E. coli, which would kill the E. coli. So we'd never get to the stage of actually being able to put the gene into the chloroplast genome. However, if we put a couple of stop codons into this gene, then it's not going to get expressed in E. coli. It also won't work in the chloroplast. As I say, this stop codon isn't used, but it still works as a stop codon. However, if we also introduced a chloroplast tRNA gene that recognized this TGA as a sense codon, then we would get specific translation in the chloroplast. So the tRNA we focused on was the one for tryptophan, because this involved quite a sort of modest change where we were just going to change essentially the codon for tryptophan, UGG, to that stop codon, UGA. So there's one change in the anticodon of the tRNA. However, we had a lot of discussion before we tried this experiment as to whether it would work. As I say, we knew that UGA did work as a stop codon in the chloroplast through early through traditional mutagenesis studies. So there must be a release factor involved in translation termination that recognized UGA. So we wondered whether that would stop it functioning as stop the codon functioning as a sense codon. And also we were going to introduce an additional copy of this tryptophan tRNA gene. So would there be competition between the native tRNA tryptophan pool and the modified tryptophan tRNA? However, surprisingly, it worked really well. So here is a Western blot. This is a control where we haven't got any stop codons in our test protein coding sequence, and we get a certain level of protein. Put 2 stop codons in, obviously we don't get any protein. But if we put 2 stop codons in plus that tRNA, then we get essentially wild-type levels of the test protein. And it's specific for the chloroplast, so that modified tRNA doesn't work in E. coli. So we don't get that suppression, we don't get that translational read-through. And when we looked at the growth curves and other factors, we couldn't see any difference in terms of the wild-type CLAMI and the genetically engineered CLAMI. So there doesn't seem to be any detrimental effect of adding this additional tryptophan tRNA to the system. So that's allowed us to make a number of highly toxic genes. So we picked 2 genes that were known to be unclonable in E. coli, and we were able to clone both of those in E. coli with stop codons and get them expressed in the chloroplast. So this gave us a way of a workaround in terms of cloning toxic genes in E. coli, and also provides this biocontainment, because, as I say, these stop codons in the gene itself and the fact that the tRNA doesn't seem to work in bacteria basically means that this is a system that will only work in the chloroplast of Chlamydomonas. That led on to some sort of interesting discussions about whether we could change other tRNAs by tweaking their anticodon so it would recognize that stop codon. And again, there was sort of discussion that this wouldn't work because the anticodon is a very important determinant for the enzyme that puts the particular amino acid onto the end of the tRNA. So the amino acyl synthetase that puts the tryptophan or whatever amino acid onto the end is using the sequence of the anticodon as part of its recognition. So we wondered whether actually the tRNA was being properly charged with tryptophan. So we wanted to sort of test that by mutating a key tryptophan residue in one of the photosynthetic genes. So this residue, or this particular position in this protein PSAA requires a tryptophan because it interacts with a key factor involved in electron transfer in photosystem 1. So this quinol here. So only if a tryptophan was put in that position would photosystem 1 work. So we created this mutant where we changed it to the stop codon, and then we introduced the tRNA gene to check whether the tRNA would actually put a tryptophan in that position. And indeed it does. Basically, in the absence of functional photosystem 1, you have this light sensitivity. So even in the presence of acetate, the light basically affects the growth rate of the algae. Whereas when we put the tRNA gene back into the mutant, then we essentially restore wild-type levels of photosynthesis, wild-type levels of light tolerance. And interestingly, we can use this tRNA directly for selection. So we can select for restoration of this mutant just using light as our selection strategy. So this now gives us a very, very simple way of selecting for chloroplast transformants. We've got this tiny little gene, tRNA gene, and we just plate the cells on light, in the presence of light, and the only things that will grow are transformants. So that gives us a very small marker, a very simple, fast method of selecting for transformants. They're marker-free, so this gene is an endogenous gene, and because it works in trans, we can actually put our gene anywhere in the chloroplast genome. This tRNA, as I say, works in trans, so we can actually use it to read through several different genes in the same chloroplast genome. So here we've actually put 2 different genes into the chloroplast genome, and both of them have got 2 stop codons, but both of them are correctly translated to give the full length protein. This second gene here is called PTXD, and PTXD is a bacterial enzyme that converts phosphite to phosphate. And phosphite is a source of phosphorus that is not normally bioavailable certainly not to any eukaryotes and to most bacteria. So if you try to grow microorganisms in a media containing phosphite, they won't grow because they might be able to take up the phosphite, but they can't utilize it as a source of phosphorus. When we put this gene in, this PTXD gene in that converts it to phosphate, you can see that we actually get wild-type levels of Chlamydomonas growth. And that's valuable because it now gives us a way of selectively growing the algae and avoiding unwanted contamination by other microorganisms in our cultures. And we can illustrate this here. This is our simple photobioreactor system. If we grow our algae in media containing phosphate, normal phosphate, and don't sterilize the medium or we contaminate it, then we get this horrible contamination with different bacteria and different fungi. Whereas this is non-sterile media deliberately contaminated with a cocktail of various different fungi and bacteria, and the only thing that grows is the Claviomonas. So we've got a very nice way of ensuring crop protection. So the only thing that grows to any significant extent is our crop, and none of the weeds, none of the bacteria or fungi. And again, because we've got these stop codons in the PTXD gene, if it does escape into any of these contaminating microorganisms, it's not going to be functional. We also thought about using the tRNA as a way of controlling gene expression, and obviously controlling the expression of those transgenes at scale can be problematic. You want something that's cheap, easy to achieve at scale, and ideally self-contained within our chloroplast system. So we thought about a very simple thermal switch where the protein, recombinant protein, would only be made at one temperature and not at another temperature. So we reasoned that we could take that tRNA that reads through these UGA stop codons and introduce a mutation into the tRNA so that if we increase the temperature, it would unfold. And if it's unfolded, then the tRNA would not be functional, not be able to read through those proteins. So we've got a temperature switch where at the lower temperature, we can make our protein. At the higher temperature, the protein is not made. So we went back to the old literature, found a paper from 1979 that basically told us exactly which mutation to make to change this tryptophan tRNA into a temperature-sensitive version of the tRNA. And it was this change here, changing this C to a U in this stem of the tRNA. And you can see that variant 1 worked really nicely, the other variants not so much, but variant 1, the test protein is made at the lower temperature. As you increase the temperature to 35 degrees, basically the protein is no longer made. And we can show that both on the Western, and we can show that with a functional assay here. So yeah, as I say, we're having fun using this tRNA in a number of different ways. And this has now spawned a whole sort of area of research looking at all 26 tRNAs and asking the question, which ones of these can actually tolerate a change to their anticodon so that they now recognize this UGA stop codon as a sense codon for their amino acid, okay? So we set up this very simple screen where basically we put a stop codon at the start of a gene construct for a luciferase NLUC. And so the NLUC will only be made if an amino acid is put in this position. Doesn't matter which amino acid it is, because the important part of the luciferase is downstream. But we have to have translational read-through. So we tested all of the tRNAs in the chloroplast and showed that something like 12 of them showed some level of luciferase production. And you can see the best performing ones are things like the serine tRNA here and serine tRNA here. Here's the tryptophan, the original tryptophan that we changed. And as I say, a few of the other ones are also showing read-through. So again, we wanted to ask the question, are these tRNAs actually being correctly charged with the correct amino acid? So we're currently doing a series of assays where we've taken some kind of reporter protein and identified a critical residue. For example, for the serine tRNAs, there's this serine here in a different luciferase called LuCP that is critical for binding of the substrate. So only if you've got a serine in this position will you actually get functional luciferase. So again, we've tested this in the chloroplast. We've set up various controls. This is the wild type control. This is where we change that serine to a leucine. There's no activity. If we put a tryptophan in that position by having a stop codon in the luciferase, and then having this tryptophan tRNA read through and put a tryptophan in that position, we get no activity. But if we use one of the tRNA, the serine tRNAs that recognizes that stop codon, then we get full activity. So we've got a nice way of demonstrating that not only can we change the anticodon of particular tRNAs, but in doing so, we're not affecting the specificity of attachment of the particular amino acid. So that just brings me finally, and I'm conscious that I've been rambling on for quite a while, but just to a couple of last slides, really sort of moving into real sort of synthetic biology in the chloroplast. And as I say, a project aimed at rewriting the genetic code, either through codon compression, codon rearrangement, or codon reassignment. And this is work that's very much in collaboration with Alison Smith, who I think gave a talk in this symposium series back in January. So, as I say, the chloroplast genome is a very simple genetic system, so it really lends itself to synthetic biology studies. And one of the things that we wanted to do is sort of think, how much more minimal can we make the chloroplast genome? As I say, we can dispense with a number of genes on the genome if we dispense with photosynthesis. So this map here is really just showing all of the regions that we've identified where we could potentially delete that region without losing the sort of core functionality of the genetic system. And if we delete all of the photosynthetic genes, then we end up with something that is about 96 kilobases, so taking out about 50% of the original size. If we just delete those regions that we consider non-essential without deleting the photosynthetic genes, then we can get rid of about 20%. So Alison's group and ours have been doing various different studies to systematically delete different regions of the genome to test our theory. So all of the filled in boxes represent sections that Alison's group have deleted. We're also looking to see if we can tidy up the genome, so refactoring. So basically clustering together all genes that have an equivalent function, such as all the tRNA genes or all of the photosynthetic genes. At the moment, they're all scattered around the genome. So again, Alison's group have done a lot of work trying to bring together all of the 25 essential tRNAs. We know that one of the 26 is non-essential, so we've deleted that. But the remaining 25, put them all together. So that gives us, if you like, a module that is, for synthetic biology, is a standalone module that we can plug into the system. And similarly, we're starting to see if we can do the same for the photosynthetic genes. As I say, we're also looking at whether we can compress the codon usage down to just 51 codons. As I say, the codon bias is quite towards 80 rich codons. And so there are a number of codons that are used, but used infrequently in the chloroplast genome. So we could reassign those. So for example, for this leucine box here, there are 6 different codons that all encode leucine. But these 4 here are relatively, or use relatively less than TTA. So we could reassign all of those to TTA by rewriting the coding sequence of all of the protein coding genes. And similarly for things like isoleucine, serine, arginine, and glycine. And for the stop codon, as I say, almost all of them use TAA, but there are 4 that use TAG. So we could reassign those as well. And that would involve changing something like 2,000 codons, which is about 7.5% of the full codon set of the chloroplast genome. In contrast, the most codon-reduced organism to date is an E. coli, which uses 61 codons, and that required 18,000 codon changes to get it down to 61 codons. So we've been testing whether codon compression would work. Can all of these genes around the chloroplast genome function if you just limit them to 51 codons rather than 63? So all of the colored boxes are ones where we've tested them by codon compression down to 51. And essentially, we see no phenotypic effect whatsoever. So we've tried essential genes, we've tried photosynthetic genes, we've tried operons. And just to give you one example, this is RBCL, which is a highly expressed gene encoding the large subunit of Rubisco. And we've made a single codon compressed version based on that scheme I showed you on the previous slide. We've also tried a combination of different codons from the 51 codon table. So 60 odd different combinations. And the take-home message from this is none of it makes any real difference to the growth and the performance of the clammy. So we're confident that actually we can take this whole genome and build it as a synthetic genome where we just use the 51 codon table. If we do that, then that frees up a number of codons that can be reassigned. And they can be reassigned in two different ways. We can either move codons around or amino acids around within the natural genetic code. So for example, phenylalanine is encoded by TTT and TTC, but we could change its anticodon so that it can now use these freed up leucine codons. Okay. And that will allow us to explore novel genetic codes and answer questions about why the universal genetic code has evolved as it has. Similarly, we can also use those freed up codons for non-canonical amino acids, new amino acids beyond the 20. So reassign those codons and introduce new tRNAs, new synthetases for those new amino acids. So this is work for the future, but basically our studies on manipulating the anticodon of the tRNAs together with our codon compression studies at least indicate that we can do those sort of ambitious experiments. Okay, so I'm going to stop there. Basically, just summarize by saying that Chlamydomonas chloroplast is a really nice symbiosis system. It contains a minimal genetic system. There are now a number of very simple tools that we've developed that allow both plastome engineering and putting in transgenes. We've shown we can further minimize the plastome, potentially all the way down to a tiny little genome of 96 kilobases, and we can codon compress that genome down to 51 codons. And that now allows us to start thinking about redesigning and reorganizing the plastome for future engineering and for making new to nature proteins. So finally, I just need to thank all of the people that's done the work. This is my current team, a fantastic group of very talented individuals, and obviously a lot of alumni that have gone through my lab over the years. Our collaborators on the genome re-engineering work, so Alison's group, particularly Pavel, the group in Edinburgh, and groups at the MRC in Cambridge, and our collaborators in Thailand on the shrimp work. And a lot of very generous funders that have allowed us to play in the lab. So I shall stop there and take some questions.
Thank you so much. That was fantastic. That was really, really awesome. There's a lot of questions in chat. Can you see chat?
I can see the chat. Yeah. So let's.
Can you read the question out loud before answering? Because the recording doesn't record question.
Right. So let's start at the top. First, do just say looking forward to hearing your talk. So Henrique asks, in the glass bead method, do you know how many chloroplasts in each cell get transformed? So there's only a single chloroplast in each cell. So it's an all or nothing thing. The glass bead agitation does kill a lot of cells. We break a lot of cells. I think we calculated once that probably only a few % of the cells actually survive the vortexing. And then, as I say, only a tiny percentage of those actually successfully take up the DNA. So it's not efficient at all, but as I say, it doesn't matter because each of the few colonies we get should be genetically identical. Edward Grayson asks, this may be a naive question, but when you make a crude algal extract, the chloroplasts are still intact, or do they get lysed too? No, they get lysed too. It's a sort of double-edged sword with clammy, because unlike higher plants where you have a large cell that contains maybe 100 chloroplasts in a mesophyll cell, it's easy to break the cell and keep the chloroplasts intact. In clammy, the cell is very small. It's only about 10 to 20 microns in diameter. So it only has the space to occupy to accommodate one chloroplast. And when you break the cell, you invariably end up breaking the chloroplast as well. Charles Rumberger asks, thanks so much for the fascinating talk. A couple of questions. What are the advantages relative to cell-free expression systems? I guess cheapness is one. So it's very cheap and easy to generate these transformant lines. And once we've made them, they're essentially immortal. We can just keep them either on nutrient agar plates or in liquid culture, or we can freeze them away. So unlike cell-free systems where you've got to sort of set them up and keep, maintaining them, then once you've engineered the algae, then using it, as I say, for something like aquaculture is just a matter of growing more algae. Second question from him was, what is the approximate cost and batch time for production of a single protein batch? Batch time, it takes us now about 5 to 6 weeks from idea to stable homoplasmic engineered strain and demonstration that it's producing the protein. The cost, the lab costs are probably, the biggest cost is the synthetic gene that we buy, which is a few $100s. So the actual cost of production is probably $500 to $1,000 if you factor in, all of the worker time, etc., etc. Louis asks, which recombinases do you use? Are they natural in the chloroplast or are they heterologous? No, this is just natural recombinases. So chloroplast genomes are naturally recombinogenic. So they're recombining with each other. I didn't really sort of talk about it, but there's a large inverted repeat on the chloroplast genome. And so those two copies of the inverted repeat are recombining with each other all the time. So there's a sort of flip-flopping. So you've got this natural RECA-based homologous recombination system in the chloroplast that we're exploiting. What is the maximum size of DNA you can be introduced? We don't know is the answer. The biggest we put in is about 15 kilobases. I know that there's an old paper from tobacco chloroplast transformation where they put in 50 kilobases of DNA intact. Several groups have tried transforming in whole genomes into the chloroplast, and this is something that we want to do. What we don't know at the moment is whether something like biolistics or glass bead agitation is going to shear, a 100 kilobase molecule, or whether we can introduce it intact. Adrian asks, would this haploid nature and the predilection for AT richness result in a higher rate of natural mutation? So the haploid genome I was referring to is the nuclear genome. So the nuclear genome of this eukaryotic alga is haploid. The chloroplast genome is polyploid. And the polyploid nature of the chloroplast genome and this active recombination means that actually the evolution rate of the chloroplast is very, very low. So there are other groups that have sequenced wild isolates of Chlamydomonas hartii, and whilst you see genetic drift in the nuclear genome, there's almost no change in the chloroplast genome amongst all of the isolates that people have looked at. Any more? I think that's all the questions. I think that's all of them, Kate. Okay.
Yes, you've answered them all. I was going to ask the question similar to the cell-free questions. I'm glad you touched on that. And I'm really excited to see the recording progress because that's a fascinating chassis.
Exactly. Yeah, no, me too. It's, as I say, we've spent a lot of time developing the underpinning tools and doing a lot of the sort of proof of concept work. So I think now, we can start doing the exciting stuff.
This is pretty exciting. That I love tool development too.
Yeah.
All right. Thank you so much. Thanks everyone for great questions,
My
and
pleasure.
thank you for fascinating talk.
Okay. And yeah, people are free to email me if they've got further questions.
And have a great week.
Okay.
Bye, everyone.
Okay. Bye.
Bye.